Good afternoon, everyone. It's the third webinar series of Asia PGI. It's really a pleasure to have everyone. And today we have an excellent talk on TB, using NGS for TB diagnostics. Now, before we get started, I'm going to sort of share a little bit of intro to set the scene, because Asia PGI, we essentially, a lot of our work is surrounding using NGS for surveillance purposes, pathogen surveillance. But really, NGS is a tool that can be used for far more than that. So just a few slides sort of setting the scene. Now, as many of us know, and many of us work together on, following COVID, there was this realization that NGS is very important for early pathogen detection. And it really strengthened our surveillance system and the use of evidence to control the COVID pandemic. Following this, the WHO did release a global genomic strategy, which is currently being implemented in countries. And some of the work that we did with many of the partners in the region have shown that most of the countries in our region actually have the capacity or some capacity to sequence pathogens and use it for surveillance purposes. However, we also know that a sequence of a pathogen can be used more than for surveillance. It can be used for diagnostic purposes. It can be used to look at whether therapeutics are still working on against new variants. It can also be used to design vaccines. Now, a little bit more. In fact, some of the landscape assessments that we did very early on through Asia PGI has shown that many of the countries, our Asia PGI partners, are using pathogen sequencing data for far more than surveillance purposes. They're using it to evaluate vaccine efficacy. They're using it to design molecular assays, diagnostics. They are using it to look at whether therapeutics are still working or to develop therapeutics, and some to look at functional immunology assays. Now, some of the work that we are doing with some of the colleagues on this call, a webinar, essentially, we're doing a regional systematic review as well through the Asia PGI network. And this is to look at the expanded use cases of next generation sequencing of pathogens in the Asian region. And this is really a sort of an effort that is together with many of the partners in our region. Now, this is really about expanding the value of pathogen genomics even beyond surveillance. So if you look at here, this is sort of a review article that was released recently. And as you can see here, there are many reasons we can do pathogen genomics for public health purposes and impact. But then again, we can easily use NGS for clinical management, as well as research and development of novel diagnostics, therapeutics, and vaccines. And if we zoom into certain prototype pathogen groups, as we can see here, TB, drug-resistant bacteria, respiratory viruses, zoonotic pathogens, arboviruses, novel pathogens, etc., we can see that in TB and several of these top pathogens, we could potentially use NGS for clinical management, so essentially for diagnostic purposes. And essentially, this is exactly what the speaker today is going to talk to us about. And just to introduce our speaker, we have Dr. Anita Suresh, who is the director of the Genomics and Sequencing Unit at FIND, a global health nonprofit focused on accelerating equitable access to reliable diagnosis. Anita leads a 10-member cross-disciplinary team evaluating sequencing solutions for policy on diagnostics and surveillance, as well as supporting genomics capacity building in low and middle-income countries across drug-resistant TB and antimicrobial resistance and pandemic threat. She is not a stranger to Singapore either. She has 18 years of experience across the biomedical value chain. She started her career at A*STAR, followed by diagnostics industry roles prior to her shift to Global Health Organization. And from that, we really welcome. It's a pleasure to have you, Anita, at this webinar, and really looking forward to hearing more about your talk, which is titled Targeted Next Generation Sequencing for Drug-Resistant TB Detection, Evidence to Policy. Over to you, Anita.
Thank you, Ruki, and thank you, Shrein and the Asia PGI team for having me on this really good webinar series. I'm excited to also continue to listen to the other talks in the series. I'm glad to get this opportunity to present on some of our work as well. Hopefully you're able to see the screen now.
Yes.
All right. Okay, so yeah, as Ruki said, my focus really today is on talking a little bit on our work on NGS for drug-resistant TB detection and really from the diagnostic perspective. And we also give a really nice introduction on the setting and sort of use cases. So there's a lot, it helps give me a nice foundation, I think, to continue from. Really, my emphasis, I'll get a little bit more detail on TB, on the sort of burden of TB. Again, there might be plenty of you in the audience that are very familiar with the stats, but it's always worth repeating just because it is so stark. I think essentially TB is a completely preventable, curable, treatable disease. Unfortunately, we still have a TB patient that dies every 22 seconds. We have over 10 million people that fall ill with TB every year, and each year, and even let's say 2022 is the most recent data that is out there, over 1.3 million people died. So this continues to remain the most infectious, sort of the biggest killer among infectious disease, even though COVID surpassed it for a while. And the problem of drug resistance is even further exacerbates this, right? So essentially, we have close to about 400 to 500,000 people, unfortunately, that develop drug-resistant TB every year. And for us to be able to tackle this, I think a big part of it is we obviously need universal access to comprehensive drug resistance testing. This, unfortunately, is not yet in place around the world. We have less than 40% of DRTB patients that get diagnosed, and among these, only 2 in 5 of those that really have the need then actually have access to complete treatment. And it's very clear that early detection and then detection that's linked to optimal treatment is critical so that we're getting people on the right treatment right from the start. The good thing is now for TB, after what, 40, 50 years, we finally have some new drugs and shorter regimens with showing very high efficacy. That's a lot of new, very recent data that came in the last few weeks, and WHO policy really supporting the use of these drug regimens, all the way bringing down treatment regimens from 2 years, which used to be the average, to even 6 months where applicable, where people are eligible for those treatments. But this means we definitely need to have ways to do comprehensive drug resistance testing, because you want to make sure we're not only protecting the patients, but also protecting the efficacy of these drug regimens so that they can be used for longer. For anyone familiar with AMR, of course, resistance is inevitable. You just want to be able to delay it as much as possible, and with judicious use of the drugs as well, and judicious use of drugs directed with diagnostics so that the patients are getting the right treatment at the right time, and we are not going through a series of treatments trying to sort of play around with saying what works and what doesn't, unfortunately. So again, a little bit on the why are we thinking even NGS here, right? So setting the context. So the standard of care for TB for drug resistance is phenotypic DST or culture-based DST. Now, even the fastest among culture, which is liquid culture, still takes 6 to 9 weeks to get the results. Now, I think anyone doing diagnosis in this call for sure, you would agree that should not be a diagnostic that takes 6 to 8 weeks. That cannot be a way to get a result when we have all these rapid tests and so many other things, so many other innovations and in progress that has been made in technology. We cannot sit back and rely on something like this. So of course, you go to the other end of spectrum, there is whole genome sequencing, which again, many of you probably are already doing for many pathogens, potentially also for TB, perhaps for surveillance. Today, this is still only effectively done using culture. You still need culture as a starting material, could be early positive cultures, but nevertheless, just because there's so much of all DNA in a sample of sputum, which is a typical sample for TB, the sputum sample, you're going to get very little of TB DNA when you use it directly and you just do extraction from sputum. And therefore, when you're doing this, when you're trying to do whole genome sequencing, you absolutely need a way to concentrate or enrich or somehow focus only on the TB bug, which is why culture becomes a necessary step. This still, which means you're adding the 2 to 3 weeks still to be able to get the result, which is why our focus about 4-5 years ago really was on targeted sequencing. Now this, you can start directly from sputum. And since you're focusing in on sequencing, amplifying and sequencing the very specific segments of those genes and regions within the bacterial genome, you're being able to target it essentially for TB right there. So essentially, that's the reason why at that point, there's obviously more improvements. We'll come to that towards the end of the presentation. But this is the reason why this made sense from looking at as close as you can get to how you can utilize the power of molecular technologies, but the comprehensiveness that you can get from a culture-based DST. Obviously, I told you some of the reasons. In TB in particular, again, more for the audience that may not be as familiar with TB, there is very, very strong genotype and phenotype correlations. It's also a very slowly evolving genome, so there's quite a highly conserved genome in that way. There's also, overall with NGS, of course, you know the advantages being high throughput, being scalable, also the lower biosafety requirements when you think about compared to culture, because there you're growing up a bacteria which was a high biosafety risk, which means you will need a biosafety level 3 lab to be able to even do that testing. Whereas with sequencing, you're only using your amplifying specific regions. By then you're just looking at DNA, so you're not working with live bacteria. And essentially with sequencing, it's adaptable to new genes and mutations. And I think that is another part, again, leveraging the power of molecular, which is to be able to hone in on the real targets, on the exact targets, to be able to say, hey, we already know these molecular targets correspond to phenotypic resistance and hence clinical resistance. But how do we ensure that as there's new resistance emerging, how do we ensure these are included? Well, something like sequencing allows you to have those in there. I'll come to this as well in the later part as we talk about the mutations catalog and so on. And overall, I think the value of sequencing or sequencing platforms is a multi-disease tool. It can't be overstated, but I think that's another big value of this that ministries of health and health programs are seeing and utilizing, saying we bought it perhaps for COVID, but we want to be able to utilize it for our current disease priorities in the country. So our work, actually, what I'm giving you is really the story of some of our project, which really goes from evidence to policy and then policy to implementation. So we had started this was based on a Unitate grant we secured several years ago. Now it feels like it was a long time ago. It was right before the pandemic. We started just in 2019, end of 2019, and the pandemic hit, but we still had to work alongside it. The goal of this seek and treat essentially was to, as you can see from name, really to sequence and to be able to get the answers for treatment. The idea was to generate the evidence and boost at least some or catalyze some in-country capacity to support the global adoption of end-to-end solutions for targeted sequencing for comprehensive diagnosis of DRTB. This consists of a few different, these 4 different outputs or parts of it. So really start in terms of the foundation for this with some of the data sets. So obviously for something like sequencing, you can imagine data is the basis, right? You want enough data to say that, to even be able to say there is enough evidence to say you can actually, there's enough molecular data that is linked to phenotypic data for me to be able to predict resistance from mutations. So that's where things like the cryptic, things like ReSeq-TB came in. And then WHO at the time in 2018 published a technical guide for sequencing for TB. And that was just purely on just the different NGS technologies and so on. But that's where they published their very first target product profile for sequencing for TB. Now that really was the basis to drive this for our grant, to be able to set it up, to invite really invite companies, invite basically innovations that are out there that have end-to-end solutions for sequencing-based solutions for detection of TB drug resistance. I'll talk to you about each of these phases and with emphasis on some of these more more data-rich phases as well. So really, the first one is really on the generating the clinical evidence, so which consisted of two parts: an analytical validation, which you can imagine you want to do that. You want to make sure that tests actually do what they claim they do. So that is something where you have a blinded reference panel, get the tests run on that reference panel. So it's a known panel, a well-characterized panel that we can unblind as an independent organization, not the companies, but as an independent body, unblind them and see that the tests actually perform. Did they identify what they say they claim they can identify? And the next phase, obviously, if they pass that, is the clinical evaluation, which was then going into a prospective diagnostic accuracy study, observation study. In this case, we did it in India, Georgia, and South Africa. Now, in parallel, we worked on a mutations catalog and a knowledge base at the WHO, which really is sort of the basis of the engine that powers the catalog. Now, this, again, because there were some bases of catalog that were already published in terms of ReSeq TB, Cryptic, and others, but it was really a systematic review of various publications, essentially, and which you really wanted to get beyond from there into a standardized list, a list of truth, a list of confidence-graded mutations. So people, companies, clinicians, researchers, policymakers could really look at, hey, these are definitely, highly associated resistance markers that are known for TB that are listed. So at a minimum, you want to be able to cover these in your diagnostic, in your DRTB test. Now, both of these, this phase, the 1A and B, which is really output 1, and then this output 2 catalog towards informing WHO policy and the use of targeted sequencing solutions. And I'm pleased to say that, of course, earlier this year, the guidelines did come out. So it was really nice to see the entire process, as long as it did take, from evidence to policy, because we did see that WHO Guidelines Development Group reviewed the evidence that we, and along other evidence from the rest of the world, really reviewed all of this in a very thorough, systematic manner with independent reviewers to be able to pass guidelines to recommend the use of sequencing. I'll come to the details of this in a bit. And then what we are continuing on 2024, really, and a little bit of next year, would be assessing some of these different implementation models of sequencing in countries. We're doing this in Brazil, Indonesia, and South Africa. And then some of the tools and activities to essentially help prepare countries for scale. So this would include cost effectiveness analysis, costing tools, as well as access pricing, ensuring these products are listed on the global procurement list, and so on. So before I continue with the data part of it, just when I keep saying end-to-end solution and so on. So just to clarify exactly what we evaluated and what we were looking at when we talked about, and what WHO is looking at as well, is really going an end-to-end sequencing solution that goes from taking the sample, in this case, the sputum sample directly, and getting to the end result. That's a report. So this includes the extraction, the library preparation, the targeted the sequencing itself, the data analysis, and then the report. Now, at that time, there were a few companies that came forward in the end. So the data I'll be presenting today will be for 2 of those that completed their review on time and was submitted the evidence was submitted to the WHO. This was the GenoScreen with Illumina combination and the Oxford Nanopore Technologies combination. So as you can imagine, none of these companies make all of these products. Some of them, like Nanopore, makes a lot of this. They can make extraction. Illumina makes a sequencer. GenoScreen makes the assays, and so on. But really, the idea was putting it on the manufacturer to say, bring together the best, sort of most validated, or the ones that you have perhaps the most footprint of, that workflow that we can then evaluate as an end-to-end lab workflow. Since it's still not one box, it is still a workflow of 2 or 3 different modules that we need to look at. Going quickly to the results itself from the analytical and clinical evaluation. So I said phase 1, which was the internal analytical validation, and there where we basically provided a reference panel, very well-characterized reference panel, both DNA as well as sputum samples, but essentially where we knew it was both for LOD to look at the limit of detection, but also for saying these are all as varied a panel of drug resistance mutations markers as we could get. because that's not something we can get necessarily in a clinical trial, necessarily in the field. It might be the more common mutations. So the idea was, let's ensure we can test this in a way, all the mutations that these products claim they can detect. So this worked well, and essentially it was clear that when a SNP is present, both these solutions were able to call them accurately, which means these products then passed into phase 2, that we could then start the multi-center diagnostic accuracy study in Georgia, India, and South Africa. This was on a trial population of confirmed pulmonary TB patients at risk or proven to have DRTB. We did over 750 patients, 750 participants across these 3 countries. And the reference was really a composite reference of phenotypic DST. As I said, it's a standard of care, and whole genome sequencing, because that also is, again, you want to make sure that results that you're getting in sequencing is also the targeted sequencing gets those as well. So for certain drugs, it makes sense to have both. Certain drugs phenotypic, or certain drugs actually the sequence overrides the phenotypic. So the idea was, as long as it's resistant based on one of 2 methods, we call it resistant. We also did the comparators of expert, of course, and the Hain LPA, since that has quite a big install base of LPA that is being done in TB labs today. And the objective assessment was for diagnostic accuracy for resistance to the set of the common 13 anti-TB drugs. Jumping right into the results, again, many of you may have seen some of this. We did present. We're happy to say the publication was just accepted, so hopefully soon we'll be able to see all of the data that you ever wanted to see in there. I'm really giving you only the high level, very few charts here. But again, to say that both these NGS solutions that we evaluated, which was the ONT and the GeneStream Illumina, they both provided accurate, reproducible results from direct clinical samples. And over 95% of these clinical samples actually produced TB sequence data on TNGS, which was really great to see, which means that as long as there was this DNA that was there, they were able to produce, those sequence results were there. And as I said, when you're able to sequence, for the most part, you do get that high accuracy. You are able to detect a SNP when it's there. So essentially, where you're seeing sequence failure, so if you see this chart itself, you're seeing the sequence failure, you focus on, say, the gray areas mainly. And that's where you're seeing it's essentially associated with lower bacterial loads. Now, we say we are defining low bacterial loads based on the expert semi-quantitative proxy. So rather than a smear, because a smear itself is a very crude way to tell. It used to be the way we'd use for molecular tests, or smear negative, smear positive, and so on. But that actually ends up missing a lot of cases. There's a lot more that you can get in terms of, even if they're smear negative, they potentially have enough DNA to be able to get a molecular test and hence a sequencing result out. So as you can see, for the most part, 91% of sediment samples of the clinical samples here produce the information on drug resistance. And for GeneStream and for ONTs, more than 94% of them produced drug resistance information. So the target failure rate was, of course, there was a 5% drug target failure rate. And there was slightly smaller in these other, in GeneStream. So I think this is more specifics on each of these assays. Again, you will see these results in the WHO GDG results as well, the entire policy document. If you've ever been interested, you will see a lot of good data in there. This is, again, a quick summary to tell you exactly how these products performed. So you'll see where they had very high sensitivity and specificity across almost all their drugs, definitely across the drugs that have been around for a while. Where you're seeing some drop off essentially is in the newer repurposed drugs, so linezolid, clofazimine, and bedaquiline. So it's still very high in terms of these. So a lot of the drugs, as you can see, it's more than 80% essentially, or more than 95%, of course, for the main key drugs. But on bedaquiline, clofazimine, while it met the target product profile minimum of an 84% sensitivity, it's still not. I mean, you want to see higher, right? Because this is compared to a phenotypic drug resistance. And the performance on linezolid is also pretty low compared to what you want, because it's still less than 50% here. But a lot of it means that then there is performance can improve as knowledge of mutations associated with resistance is expanded. So it could either be that these products don't have those mutations either captured or reported on in their assay itself. So, in the final reporting stage, in the bioinformatics stage. Or more or likely for some of these newer drugs, and which we haven't even looked at for like predominant, the drug targets aren't even known. So then we don't even have the information in the world, in the knowledge base of the world to know what these drug targets are, to be able to say with certainty that these mutations that are associated with resistance, and hence we are finding these mutations. So with ONT, it becomes even clearer. So you'll see all of these for the top drugs are definitely high sensitivity. You're seeing the drop off very similar to linezolid. So which means it suggests that maybe that's where you can get today with a molecular test. So that's something that is being looked into further. But you can see very low, almost no sensitivity here, no coverage for clofazimine and bedaquiline. So again, it was clear that sequence data was produced, but the performance was, they did not call any targets for these drugs essentially in their final report. So which means we suspected that the performance could actually improve if there's an updated mutations list. So if they update their software to reflect more targets, because they've already sequenced it, they already have it in their targeted NGS in their assay, all they need to do is to make sure their bioinformatics reporting actually calls all those SNPs as well, or all those mutations as well. So putting together all of that, there's a lot more detailed data. If you're interested, that the publication will have all of those details for you to look at. But the key here was essentially taking that evidence to WHO, very, very rigorous guidelines development group review in the last year. And it took several months to get the final guidelines published, but at least there was a rapid communications right after saying this had been reviewed and this is positive recommendations. Essentially, you'll see the final guidelines gave a class-based recommendation to use targeted sequencing for DRTB diagnosis in two situations or two sort of cases in terms of in the target and sort of the populations that you could use it in. So one being those with bacteriology confirmed pulmonary TB, and then those with rifampicin resistance pulmonary TB. So obviously as a reflex from a molecular rifampicin resistance result. So that's usually more the common case because countries have either things like Expert or Abbott or BD or Roche or even Armol Bio. So there is essentially there's already that test that they're getting, that result they're getting in a way closer to primary care. Now, what do they do with the reflexing from there when they are seeing resistance, or there is a high risk of drug resistance, even if they didn't get drug resistance or they necessarily didn't get a clear answer for drug resistance. That's where I think there's a room essentially, as the language says, room to reflex to TNGS to be able to get resistance results for all of these other drugs. Again, there's always caveats, and it's WHO policy, so definitely lots of caveats that will be in there. So that's definitely something to be read as a whole in terms of why it's conditional, what does that mean, and so on. So of course, there's as you saw, there's certain drugs where it's still suboptimal. Again, these are new drugs. There's not enough data that's out there. There's in some cases not enough resistance that's out there, which is a very good thing. We don't want more resistance to these new drugs, but over time, unfortunately, there will be resistance that emerges. So that's where I think we want to make sure that a technology like this can keep up with that. We can quickly update, get those as the knowledge improves on the resistance associated with resistance associations, mutations associated with resistance. We are able to link them back to an assay and get the test to actually report on these mutations as well. Other limitations, obviously, it's still not enough evidence on the cost effectiveness. So that is something I know different groups, our group as well, but other groups as well, I think even within Asia PGI are working on, because this obviously needs some real world cost effectiveness or costing data. We did have data that was collected during the clinical trial, but of course it's a very highly controlled environment. So that doesn't necessarily reflect the real world cost analysis. Since I was talking to you a little bit already on the positioning on where does this fit in the testing algorithm, again, this is probably most relevant only for people in the TB, specifically in the TB world, and in that case, they'll be very familiar with this. So this will be too basic, but essentially to say that where there is obviously always like where do you fit this in the algorithm, right? So there is the TB testing algorithm is pretty complex, depending on what is available, depending on cost, depending on the epidemiology, depending on the health burden in the country and the health system setting and so on. So you'll see there's various nuances to the typical trend. It's not, we can't say there is one algorithm that fits them all, one algorithm that the world follows. But it's really based on all of these factors that I mentioned. But overall, I think this is a recommendation, as we said, obviously, as we're looking at all persons with drug resistance or TB, when they can treat, if they're obviously eligible for treatment, get them treated if they're not from MDR. If you otherwise basically essentially collects these samples to be able to do targeted sequencing. And culture, this culture is still recommended in some cases, but the idea is you could do NGS essentially instead of in place of, or rather than culture for most cases, because you're going to get this result within a couple of days, as opposed to this, which is going to take you 6 weeks. So that's why countries are very keen that they are actually set this up, because if you can get a result earlier and we put the patient on the right treatment earlier, that will make a world of difference in terms of the TB management and the programmatic use of this test itself and the applications for treatment. So that's more sort of where this falls, where they have a few recommendations on how this can be used. But the idea is obviously it's up to countries to decide where it makes sense. So as we work with countries on implementation, and as many of you are working on it, or partners like Asia PG are working on it, this is something that will become relevant. We can't really prescribe this is where you should fit it, but there might be cases where in some settings it might make more sense that all cases, essentially all samples are reflexed, all samples that are RIF resistant are reflexed to TNGS. In some settings, it might make sense with the lower DR burden, all TB positive samples are reflexed to TNGS. Now, this is not obviously the more common one. This is being done in definitely in low burden settings, low DR TB burden settings. Brazil is, for example, looking at their, in a way, implementing it, doing an implementation pilot of this. Of course, New York and UK and Netherlands and so on, like the essentially low burden, high income settings are definitely doing that, following that method, because of the risk tolerance is obviously much lower in terms of drug resistance. So you want to make sure every TB positive sample gets the full comprehensive drug resistance profile, and hence sequencing is being done for all TB positive samples in those cases. Now, I want to make sure I've taken quite a bit of time, but I think I want to move on to this because I spoke quite a bit. I think we can't actually separate the whole mutations catalog and sort of the interpretation story from the clinical evaluation aspect of sequencing. Really for any molecular test, but more so for sequencing, because again, you're getting so much data from it, as you guys know. But it's so important that we know exactly what to do with it. It's not data for the sake of, it's really every single thing that you collect should be of use. And in this case, truly of use to actually direct treatment, right? So this was essentially, that's why a very important endeavor. For years, people have been asking, when I say people, it's companies as well as scientists and essentially the TB community has been saying, what is this list of truth? Is there a standardized list? As many people have been publishing, many databases that exist. How do we get to that one list of truth that actually is that master list or standardizes, or these are the mutations, these are the targets that one should be evaluating for, knowing that this list should get updated over time as new resistance emerges in the field, as it will. So essentially, the idea was this was an opportunity with this grant to actually say we'll work with WHO to set this up, because again, we wanted to not set up a brand new system. The idea was what can we utilize where there's already maybe data sharing, maybe things that are already being sent, say, from surveillance. So this is where the surveillance data, because drug resistant, because TB is obviously in a way a reportable disease, and hence there is annual or at least regular surveys being done, DRS as they call the drug resistance surveys that are being done across the world. And every country in different frequencies or the cadence would differ quite a bit. It could be 5 years, it could be every 8 years, it could be every 2 years, and really it differs, but really depends if when the program can manage it alongside their actual direct daily programmatic clinical needs. They're able to do this DR surveys to share that data, and that's how you get that whole WHO report that you see every year on TB has a lot of these summaries. And there was all of that data is there, but a lot of it is sitting there. A lot of it is in the back end. A lot of it is sitting there where only 1 or 2 people at WHO can look at it. It really doesn't help everyone else in the world and doesn't help the countries in a way that way back or the programs back. So this was really an idea to say, how do we see how we democratize it in some fashion to get to a stage where we can actually get a catalog. And for this, we need all of that knowledge base, right? So all of the resistance data and hence all of the sample data and the data from TB samples that are both culture results as well as the sequence results, correlated, associated sequence results. I won't bore you with all the details. Again, there's 2 fabulous publications we've had now. We have the version 1 catalog in 2021. Happy to say the version 2 was released end of last year. Essentially, what you need to know is that this is a catalog of all the resistance-associated mutations in TB. This allows you to do a very standardized analysis and interpretation of the data, so you can predict drug resistance from sequence information. So this is something you as a lab user may not necessarily use directly, but certainly the companies that are building a TB assay are using this as their blueprint to design their assay. In addition, I guess as researchers, as there's surveys conducted or projects conducted on sequencing with samples that are there, that data also goes in here because that's also then valuable to say, what is that? If you're starting to see a mutation emerge in a particular region, over time, you're going to see more of that, right? But essentially the frequency of that increases over time. So the earlier you get it into this database, the better. So there is some capture. Even if we say, hey, we've seen about 5% of 5 occurrences of this, even let's say even 5 occurrences, people are flagging it in some cases. But we want to have enough statistically significant data to actually push it up to saying definitely associated to resistance, but at least you'll start seeing it here as something like potentially associated or a low confidence association. And over time, that could actually get bumped up to the next level. So this is just to say that as countries are building their own databases, that this is one that has had this catalog is something that helps in the design of new diagnostics. And that is going to be critical, be it molecular PCR-based tests or be it sequencing-based tests, to ensure these tests keep up with resistance, emerging resistance and the new drug regimens, because these treatment regimens are being rolled out actively. And over time, as these get more widely sort of disseminated and wide uptake, you're going to see, unfortunately, some of this resistance emerging. So you want to make sure then we have that data is going in here that's already captured. So the tests also keep up to make sure we are able to continue to get the right treatments to people. I won't go into more. I'm happy to answer more questions on this, just to say we have a version 3 in development, because of course we know this is never we can't just stop it when it is never static. We sort of need to keep changing it. You all know from COVID and other things, it's a lot more dynamic. Thankfully with TB, it is not so dynamic. So it gives us some time. It is a very slow growing bacteria, thankfully. So we also slow to develop mutations in that way. A little bit more on that, and I apologize if there are non-bioinformaticians here, but I'm hoping that at least people with interest in bioinformatics will find this a little bit interesting. But essentially to tell you that, and I told you about improved performance, right? Because that you can tell me, Anita, what makes you think it'll improve. So here's one way we have a way to actually show you, at least from evidence, just from the catalog. So we know that when the sensitivity is low in some of these products, we know that a sequence data is generated for these targets. But performance, so sequence data is generated, which means if these targets are actually, the calls are actually made, then the performance can actually improve. The sensitivity would actually be higher. So I'm just showing you a comparison of the catalog version 1 and version 2. Don't worry, but so these are the sensitivity versions of it. These are obviously matching, they were high enough already. But you see for these drugs, we had nothing. We didn't even have any data corresponding to resistance in these drugs in the first version, because that was obviously 2021. So the data was really any data that came before 2021, which means these drugs weren't really even being utilized in treatments. But after that, even WHO did a very specific call for data, for resistance data on these drugs from the field, from any clinical comes and so on. And based on that, we actually increased. There was a lot more data that was added. I showed you like it was another 15,000 samples and so on overall. You could see suddenly you're seeing a 78%, 74% sensitivity for these two drugs. So any product that utilizes then version 2 and sees those resistance and actually adds those mutations, at a minimum should reach this sensitivity. They could be higher if they choose to use more, but at a minimum, they can reach this if they ensure they have all of these mutations that are listed that were identified through the statistical analysis in these catalogs. So that's essentially what I'm going on to describe here, where there's essentially you can see additional mutations, increase in sensitivity. So as long as products have these regions amplified in their PCR aspect of it, it will definitely, all they need to do is report on it because those regions were already sequenced. Now, there's going to be certain drugs like predominant and others, or even linezolid, as you can see, it's still pretty low because there are certain targets we don't know yet. There is also not that very high resistance for linezolid overall in the world. So that makes it harder, obviously, to make those correlations, associations. But again, this is something we like to say that as the knowledge improves, our knowledge of mutations improves, the performance can also improve because these can then be captured in the catalog and hence in assays and products that are developed based on this. So moving away from all of that data stuff, in the end, yes, the product we say it has worked, and there's WHO has given its sort of stamp of endorsement and recommendation for utilization, but that's not enough with sequencing. As many of you know, since you're using sequencing for various applications, there's a lot more that goes into it. So some of the other work, again, there's just a snapshot of things that we are working on that might be of interest. Obviously, we have tools for assessing country readiness for NGS implementation. In fact, the TB programs should be getting a couple of more surveys soon from the WHO on some of these. These will in a way help the countries, but also help programs and also help in a way in terms of resource mobilization, right? Funding and funders who are interested in supporting capacity building to understand what is the readiness, what is available already in country, what else is needed if a country wants to pivot or leverage their existing sequencing infrastructure for disease X or TB, so on. And then beyond that, one more thing I would highlight is this whole thing of the diagnostic network optimization. This might be familiar to some of you. This is very much something that's utilized for things like expert and so on. It's really to optimize where diagnostics are placed and sort of the distance from where diagnostics are placed to the number of samples that you can access there, and the number of patients or people that can access that testing facility. So it's really optimizing in terms of how many of these facilities do you need versus where is the cost benefit? What is that trade-off that you need to do? And where is it more effective, cost effective to have, say, fewer number of devices or diagnostic devices, but just that there has more samples can go on them. So for sequencing, this is obviously something makes a lot of sense, especially because it is truly can be used not just for TB, for everything else. You don't need 50 or 100 sequencers in a country. You might need the few that you already have to be better utilized and more optimally capacity utilized for more of your priority diseases. So that's another tool we're working, at least testing it out with Brazil and a couple of other countries that are interested. There's all the training and getting products on the list. And the costing tool is one I wanted to highlight. Again, you might be familiar, you've seen this already. WHO has published a costing tool that we partnered with a number of organizations on this one. We're working on the TB version now. We're testing it out with a few partners. Essentially, it's an Excel-based tool. You'll see it on the WHO website, initially developed for SARS-CoV-2, but takes into account all of the factors, right? It's not just in terms of the reagents, consumables, equipment. Obviously, the basic, you need that pricing for sure, but also in terms of personnel, training, facility itself, infrastructure, any QC, QM needs, transport, and so on. Because this will really allow not just for in a way, helping the sites in a way budget for what they need to forecast, but also help with planning in terms of then for Global Fund and others, for them to also be able to say how these will be, to understand how the funds will be effectively utilized by the countries, by the recipients. Towards the end here, just to say where it mentioned this programmatic implementation. So it's one thing to do a clinical trial, generate the data for evidence to get policy, but obviously the more important thing is then translating that policy to implementation, real life implementation, and really understand what are those challenges, what are the bottlenecks there, or what are things that need to be further optimized or worked on. We are working with Brazil, Indonesia, and South Africa on this in different capacities. So essentially, the operational assessment that we're doing in terms of their implementation, and as I said, different algorithms, how they're looking at it, or different settings, and so on, different scale that each of these countries is doing it at. Happy to give more, but these are all ongoing projects right now. We also have a larger implementers network focused on the TB work to essentially be able to share some of these learnings, because there's a number of countries either implementing or doing an implementation pilot and wanting to then run it programmatically. There's countries like Eswatini, Bangladesh, and Kyrgyzstan actually have, either it's informed or there's policy recommended for the use of sequencing for programmatic implementation. That is national policy, not just the WHO policy, but national policy, which is really great to see. And this is more an example, since I'm also presenting it in Singapore, just to more talk about New York State as an example, where they in 2023 essentially approved the use of targeted NGS for rapid detection of DR drug resistance in respiratory specimens. So where they were using whole genome, but they really pivoted to targeted sequencing based on that improvement they were seeing in turnaround time and just for overall performance and all of those good positive benefits they could see and they could actually prove. New York State being one of the highest sort of the most notorious in terms of being able to get through, get an approval through. So this was really great to see that they are now utilizing TNGS as part of their statewide implementation. Well, last couple of things is more some sort of things that are more out there, right? Because we talk about this as if it's a standard one size or a one methodology or every country, the national program will use it, it's centralized, so on. It's obviously multiple different models that might fit different countries. And in some countries, a combination of these models actually. So obviously centralized, the most obvious one, which is there's an established sample referral network might exist in countries. You see this very well in South Africa, for example. It's super well set up already. The health system is very centralized. This allows then for them to actually centralize a lot of these equipment so that they have very high throughput sequencers, high throughput PCR machines. So they have the highest good expert machines in there. So they've been able to actually consolidate, be able to efficiently transport samples and get the results back to the clinicians and patients rapidly within a very reasonable, very good turnaround time for this to make sense. This may not work in all settings based on geographies and funding and setup of health systems. But this is a model that definitely allows for use of these higher throughput sequencers and get the maximum where your cost is obviously going to be lower per sample. Decentralized is a big one with things like Nanopore's offering and other upcoming tools that are offering more of this sort of easier to use. You can do it in any setting sort of setup. You still need some work in terms of processing the sample, but it really allows you to go really closer to that patient or closer to the facility or closer to primary healthcare where needed, where it makes sense. Obviously, if you're looking at outbreaks, if you're looking at something quick where you want to be able to get result and can actually do something about that with that result, it makes sense to be able to consider this, and countries are looking at a combination of this. The service model is something relatively new, but for TB, but it has obviously been out there for many other things, for everything else. But for public health, this is not as much utilized, but there is definitely an interest and a potential to utilize this. India has examples of two companies like Haystack Analytics and MedGenome. Essentially, the samples can be sent out. It's either from the program, from hospitals, and so on, where they are the sequencing service providers. I know Singapore has similarly one of them as well in the list. These could be core sequencing facilities in in large health systems or academic groups, or they could be in a way commercial providers like this, where because they can really lower their costs, right? They can optimize their costs. They can put on whatever samples, as many samples as needed in one run. So it saves essentially the individual labs or the groups from setting up their own sequencing facility. It is a very, very high capital investment expenditure at the start to set up. So that this allows you at least initially to start getting sequencing results, utilize it for your program, see how well that works and integrates in your system before making that call for investments or actually putting in that request to do that, because this gives you a very nice low-cost optimized way to run these. Okay, almost done here. And I said some of the improvements to workflows. There's so much there. I'm sure I haven't even said, I know this question's coming up and I haven't seen them yet, but essentially to say there's a lot of these, obviously nothing is not perfect yet. Otherwise we would have been using it for many years now. And there's still things to do here, things to optimize. But I think getting the policy out was important. Having countries having many of you guys have utilized sequencing now for COVID, for anything else, for mpox and so on. And so then there is actually that capacity that exists, that training, the personnel that exists with sequencing. So you want to be able to utilize it, but to build it on for TB, there are still some things that some gaps or some improvements or additions that need to be made to truly make it viable as a large scalable solution. So we said one of the data analytics aspects, I won't even talk more about that, but here just this little table is an example where, again, it's yet unpublished, but I can tell you that essentially for ONT, we saw this essentially the original pipeline that they had. And you saw that very poor performance. You saw that already in that little table where you saw Miracle and Clofazimine, there were barely anything. But with updating their pipeline, so updating their software, their bioinformatics, just based on their mutations catalog version 2, they were able to increase this basically to a 70% or 66% for Clofazimine in terms of sensitivity. So that's really shows that power of being able to utilize the bioinformatics, just make that change in the reporting to be able to improve the performance. So this is something I think as manufacturers look at. This also allows WHO and other bodies to make that policy change based on, okay, so they can do a quick policy review to update these so that we can update our recommendations as well, saying these products also meet this criteria. There's more in there, but I think some of the other things you can imagine are automation, right? Obviously, it's like I've taught you about 5 different things. It's extraction and the targeting, the PCR itself for amplification, the library prep, the sequencing, and then the reporting. But there is automation that's possible. There's people looking at combining the sample prep and library prep together, almost eliminating the need to do a separate amplification, doing all that in one tube and so on. So there's a number of NIH grants in place that are actually actively working on this. So there's things that essentially in works to essentially say, how do we reduce, basically make it more, simplify the process, reduce the number of hands-on steps, reduce the just overall number of steps, around number of tubes and so on. So it's good to know that there is ways to go there. There's a lot of these improvements have been done for oncology, done for so many other spaces. So I think for TB, other areas, other diseases where it becomes more of a LMIC problem. I think it's so important that we are able to utilize all those innovations or improvements that were made there, those positive externalities, the benefits we can use for diseases like this, where we can very quickly accelerate. We don't have to go through that entire curve of struggling to see how we get that. We can actually leapfrog into something that makes sense in the settings we need it for these very high priority diseases in these settings. That I thank you, and really thanking our work is really made possible by our donors, our country partners, our team. And a quick shout out on the little the union conference. I don't know if some of you might be there specifically for the TB folks, but essentially there's a number of sessions we'll be at, and I know Asia PGI is hosting one as well, which you highlighted, but several sessions there. Happy to, if you want to contact me or via Rookie, just let us know, and if you'll be there, we'd love to have you stop by as well. Thank you.
Excellent. Thank you, Anita. And for the audience, please do, for whoever's going to be at the TB Union Congress in Bali in November, Anita's going to be there, a couple of colleagues from Asia PGA Secretariat's going to be there. There's an Asia PGA workshop as well, and several that FIND is organizing. So definitely attend. We will potentially also send around some information after this webinar as well. I think we have a few minutes, Anita, for questions. That was excellent, very detailed, very sort of in-depth presentation, and you really took us all the way from design to policy to implementation. Let me see, there are a couple of questions in the chat box. There's a quick one, and I think this is something that many of us wonder. So Sashika is asking, why do targeted sequencing, why is targeted sequencing required for diagnosis of TB? Can't we use qPCR to determine TB faster? Won't that be reliable?
Yeah, and I can quickly answer that. Yeah, absolutely. Definitely use PCR for TB detection. That's not a question for TB detection itself. Here we're talking about comprehensive drug resistance, though. That is many, many targets. Today, there is no PCR test that has all those targets. Obviously, there's tests that come over time that have those. If they can be, you can make these, cover all of these, that makes sense. But there's an inherent limitation in PCR because there's only so much targets you can fit in there. And it's also, based on very specific, SNPs and targets, right? It's not going to, whereas with the sequencing, they cover the entire genes and multiple, as many genes as you want, really. So that's why I think that comprehensive aspect is there. So of course, that's why you have an expert TB today from GeneXpert from Cepheid that has a RIF resistance. You have even their XTR, you have the MolBio that has a RIF and an INH. So those would make sense. Those should definitely be your first call, your first test that you'll use. But this is for anything that if they are determined resistant there, what do you do then? Because this is for the reflex from there. Absolutely.
Thank you, Anita. And how many, this coming from someone who's not a TB biologist, how many resistance genes are we talking about roughly that's characterized in that catalog, WHO catalog?
So I think we evaluated over 200. I mean, they have a total of, I'm trying to see how many genes. I think it's, sorry, I'm just going to go to my slide there. I think there are too many numbers in there. Let's see. So 38,000, I say we had looked at about 1,500 or so genes, but I think it's about 100. It's within about 90 genes that cover most of the known mutations. Okay. And yeah, so that's about the read, and that's really hard to cover in and some like these a PCR test cannot cover it because it could be mutations in any of that entire gene.
Exactly. Actually, related to this, there's a question, very interesting question here. It says, thanks, Anita, very insightful presentation, especially for the New York Public Health Lab switching from WGS to targeted NGS. What were the main considerations for these, specifically in relation to use WGS for epidemiology-linked studies? I had the same question, actually.
Their consideration. So this was for clinical use. I think that's where they saw the value because they were able to get coverage for pretty much all the targets they could act on. So you're right from an epidemiology, if they're just doing, in a way, surveillance to understand all the mutation, that makes sense. But for a country like New York, country, but New York is a country. But essentially for a place like New York, where the volumes are getting and they do get the diversity of samples, obviously the resistance they're getting could be, even though it's so rare, they are going to get a lot of diversity. So they are still doing WGS in a way for surveillance purposes. But I think for the clinical use, they just found this more cost effective just because they have all the targets that you can actionable, that are clinically actionable essentially in that. So there's that balance is still there. I think they still continue to do WGS. Yeah.
So that's very important. I think that proportion of the samples still go through WGS, right? Especially for those new mutation that needs to get into the catalog and so on. Is that correct?
Exactly. Yes.
Okay. And that proportion that needs to go through WGS, are there guidelines for that, or are each country trying to figure out how they would go about it? Or does that depend on resources?
So yeah, a little bit. So there is the whole surveillance guidance that's out from WHO, that an NGS specific surveillance guidance. So that links to both the For TB, specifically the DR-TB surveillance, there's some criteria in terms of percentage, what is a reference, what is a strategic sampling they can do, and so on. So there is guidance out there, I think, in that, in the surveillance guide and along with the NGS surveillance guide.
Okay. I have a very long question from someone in our team. Thank you for the insightful presentation. I appreciate the detailed recommendations and scenarios for integrating TNGS for existing diagnostic workflows in LMICs. Could you elaborate on how you plan to address challenges related to long-term funding, local capacity building, and health system integration to ensure the long-term sustainability and scalability of TNGS for TB DST in Asia? How feasible is it for doctors in LMICs to use this technology for bedside diagnostics? And what support of training would be necessary to make this possible?
Okay, the first question answer is actually easy. It's Asia PGI. There would be partners like you, Africa PGI. I think we're looking to implementers like you, in a way, implementing partners like you guys, to really say how we can support at least things we are learning. Kind of say, because you guys would have then feet on the ground, or the way it's set up is more that way. So I think the idea is definitely to partner with the implementers that already exist to say what we can provide and sort of support you to get to that, because you'll have the wider view as well of not just TB, but across disease. So I think that would be the most sort of effective way, I think. And of course, working with the countries in a way, as you guys do directly, to see their needs. Again, it's not going to be sort of saying, okay, this is what you should do, but really what are your priorities and how do we build from there, from their needs.
Right.
Mm-hmm.
And you know, one of the things that we were, so essentially, especially analysis, I think there are several questions on analysis, especially one that someone's asking, are there automated web-based tools for data analysis for WGS of TB? TB is a gigantic genome.
Yeah, if it's for TNGS, of course, all these products actually have their, if you use targeted sequencing, they have their automated web analysis. So it's either web-based or local installation. So they come integrated with that. So I definitely recommend that's part of their product, right? You should use that. But for WGS, then if you're doing, there's a number of, trying to see which ones, but I can put that, I think when you send back the answers, we can put a few of those. There are some of those free ones also available, pipelines that are available that have combined a few of them. I think there's one from trying to get his name, Stefan Niemann's group, who they have a number of these pipelines that have been well utilized, I think, for WGS, and they're free to use, open source.
Okay, great. There's another question on, would you be able to elaborate on the access pathways and processes for non-WHO approved TNGS methodologies to be incorporated in the workflow of TB-DR diagnostic labs? Not exactly sure what that means, non-WHO approved.
So essentially, I think I understand where they're coming from.
Okay.
But essentially, the WHO approval was for a class of technologies. So that actually is a very good thing, because I think that's the whole point of if we had more products that were there would have been more products, data, their thing. So you would have seen the latest one is a call for data that WHO has sent out. And if you missed that, definitely, and this is obviously companies can't be sending the data, but if companies have sites that they worked with that who can submit data on their behalf, not on their behalf, really, from projects and studies. WHO has done the call for essentially the next set of data. So what WHO typically does is they do this guidelines development group when it's a new test, right? A new class. So first in class is what they did. So this was a first in class, and then they'll do the sort of best in class that was done. So anything that follows now will be a less onerous review. It'll be a technical advisory group review, which allows for, hey, we already know the basis. This is sort of the foundation we have. This is the baseline. So do any new tests that come, are they at least as good, or are they even better? So that would be a basis they would use, as they did for the expert. So Gene Expert on its own was there, but then they had this whole centralized, they had multiple products evaluated. So I think that's the similar basis here. So there are products that are, I mean, I would welcome much more products in the market. I mean, we only have two. So any more that are out there that are looking. So if there are people, I think the call for data from WHO is one. So Ruki, I'll make sure I add that link in there as well. So, and companies that are looking for that, I mean, to see how to get onto that pathway, they would need to make sure they can reach out to WHO as well, or if they have sites that have data generated that they can just submit, or publications that would really be helpful for WHO to then consider them in the next round, which should be within the next 6 months.
Right. And that would be great. Actually, Asia PGI, we are working some of the sites to test out the MGI provided TB resistance kit as well. So definitely, you know, those colleagues who are interested, reach out to Anita, reach out to us. We can connect you with Anita. Actually, we're sort of over time, but there's one question that I think would be quite interesting to answer, especially in this age of AI. So colleague says, greetings from Sarawak. Thank you for the presentation. Can CXR data from TB cases be in any way, such as using AI approach, be combined with sequencing data, be helpful or relevant in the diagnosis of drug-resistant TB?
So the AI is definitely being used with the CXR data, right? So there's a lot of that. There's an entire CAD platform, multiple 50 over products that went through. Even WHO has approved those policy. I don't think drug resistance is something they can detect today. I mean, it's more of yes or no, right? TB yes or no, but just in a much higher, much more reliable, much more sort of reproducible, all of that with AI-based approaches. Because again, the training set has been there, so they've learned. So far, we have not, with X-ray is not enough. It's going to only tell you something is there, right? Or it's going to tell you even where and how much, but not drug resistance is not something there. But in combination, absolutely. Right. And so there's AI utilization in sequencing. I won't even come to that because will bring out my nerdy self out. So I won't say that. But there is that utility from using generative AI for this as well. Because again, it's about data, right? And if instead of having 50 experts look at it and do systematic reviews, I should be able to use generative AI to actually do that systematic review across all data, all publications, all literature, you know, on a regular basis to pull that. But yes, absolutely. I think a combination of that would be great because you're getting the TB detection in a way and looking at for drug resistance.
And actually, one last question before we end. How often, because there'll be new resistance genes popping up, right? New research. How often does the WHO catalog that FIND works with closely be updated? How often can we expect a new catalog?
I think it's really related to, so what we're working on, we've squeezed in as many as we can while we have a grant, because there needs to be a sustainable process to continue doing this. There are certain groups that are interested in picking that on and working with WHO. But what we have done is set it up to actually make it as smooth as possible and also as automated as possible to actually lower all those costs. The initial costs were very high because we had to get all that data together. So now we're only looking for, so each year you'll see that call for new data. There's going to be less and less of that, hopefully, unless there are many new drugs coming in the market, which unfortunately doesn't happen for TB so often. I think over this next version 3 would be an important one. After that, we see maybe every 2-3 years, perhaps just a supplemental in a way to add, like, is there anything else anything new that's emerged that we need to capture? Because otherwise, pretty much for most of the drugs, we can say almost 100% of that resistance is captured. But for the newer drugs is where that gap is. And so that will take at least a few more iterations. So right now, I think our rate has been like every 2 years. So I think next year is where we're looking at a version 3. But let's hope after that we should see how much more, because if we can cover all the existing drugs, that's already pretty good as we can get, right? And then over time, there's going to be more resistance emerging.
Yeah. Awesome. Thank you, Anita. I think we've come to the end of our time, and really, really looking forward to, many of us I won't be there, but many of our team will be there. And I think many of the TB colleagues on this webinar will be there in November. So please, everyone, go ahead, reach out to Anita in November if you're there in person. Do reach out to her by email as well if you have questions, or the Asia PGI Secretariat. Thank you, everyone, and have a good day.
Thank you.